Hi, this is Science Hangout from Science Mission. We have Dr. Kenneth Kosik with us today. So he's a professor, uh, Harriman Professor of Neuroscience at University of California at Santa Barbara. So here are some of Dr. Kosik's achievements. He has published hundreds of research articles in peer-reviewed journals. He is in many research review committees and editorial board of several journals. In addition, he has won several awards. So I'm not going to go in details because it may take the whole hour basically. So I'm going to stick to a few of them. Then let's go and talk to Dr. Kosik. So Dr. Kosik, in 2009, he won City of Santa Barbara Innovation Star Award. In 2002, Premio Aventis Academia Nacional de Medicina from Colombia. 1999, Dr. Kosik won NASA Group Achievement Award to Neurolab Science Team. In 1997, he also won Ranvel Capito Medal, Argentine Society of Neurochemistry. In addition, he won Zenith and Temple Award from Alzheimer's Society. Welcome, Dr. Kosik.
Hi there. Thank you.
So nice to have you on Science Hangout today.
Yes.
So can you please talk about your background, education, and some of the research projects you are working on?
Absolutely, yes. And again, thank you for having me on. Well, my training, I think if you were going to categorize my background and training, I would be in the category of what people call physician scientist.
Mm-hmm.
Although I have only trained as an MD, I don't have a PhD. My work has evolved into a bridge between medicine and fundamental research.
Okay.
I have sort of a foot in both fields and enjoy them both.
And why did you plan on becoming a neuroscientist, neurologist early on rather than any other medicinal subjects?
Well, I've been fascinated by a lot of subjects through medical school. I was fascinated by tropical medicine and psychiatry, emergency medicine. There were many fields that appealed to me.
Okay.
But when I got to the end of medical school and had to make a decision, it was really clear that the brain was the most compelling.
I think you made a right decision, I guess.
I think so. But I don't know what other paths would have led to.
Yeah. And can you you had a long career so far. So can you please talk about a little bit more about some of the people who supported you, mentored you, or coached you along the line?
Sure. Well, when I started my career, I had applied for a funding as a postdoc. And at the time I was a postdoctoral fellow. I just finished my neurology residency, and I didn't have any laboratory training at all. So it was very hard for me to get into a lab, even though I knew very much that's what I wanted to do. I did ask Dennis Selko at Harvard if I could join his lab, and he reluctantly agreed with the caveat that I could come if I got my own grant at the time.
Okay.
I don't blame him one bit because I had no bench skills whatsoever. So one very lucky break for me was I did write a small grant to the Huntington's Disease Association, and it got funded for a very small amount of money. Funded by Nancy Wexler at the time, and she really, I think, was very instrumental in getting things at least started.
Okay. And in addition to your work, do you do other things like hobbies?
Well, I enjoy my work immensely, so I sometimes consider my work the hobby. You know, people have said that if you enjoy your work, you'll never work a day in your life, and that's the philosophy I follow. But I do enjoy traveling. I enjoy biking in the mountains around Santa Barbara. There are things I really enjoy doing a lot.
Okay. And can you please tell us about your lab people, and all those things?
I have a lab of mostly graduate students and postdoctoral fellows. It runs over the years between 15 and 20 people.
Okay.
And we have a lab where I think there is a fair degree of sophistication among the graduate students and postdocs. They really are, I think I learn as much from them as they learn from me. They often come to the lab with a lot of skills and knowledge. And I often think of the lab as a place where I'm creating an intellectual atmosphere more than actually specifically defining projects.
Okay. And how do you motivate them? Like, is there any technique you use, or like they are self-motivated?
They are pretty self-motivated. This is a very exceptional group, and I think when they come to the lab, they fully realize that the only way to make it in science is to work hard. I don't have to do too much motivating.
Okay. And how your interaction with them? Like, you meet them quite often, or like it is just you go into the lab and talk to them? How does it go?
All of the above. We have our regular group meetings in which each of them is responsible to present to the whole group. I very much enjoy more ad hoc informal meetings of wandering into the lab and going over to someone's bench and just chatting. And also when people get close to a publication, or they're still trying to define where they're going, I also will meet with them individually. I sort of like to think of one thing I like about the way science works, that everybody gets a chance to present. I often think that this reminds me a lot of improvisational jazz, in which you go around the group, and then there's a little solo from the bass player, and there's another solo from the pianist, another solo from the saxophonist, and then they all play together. And lab people really have to be able to perform solo as well as be able to perform in a group.
Yeah. Okay. And can you talk about a little bit of your projects, and maybe you can present meanwhile? Sure.
The projects are all in the area of neuroscience.
Okay.
They span from very much disease-related projects, very much oriented toward neurodegeneration and Alzheimer's disease. There are other projects in the lab that are a little more basic, having to do with aspects of stem cells and how stem cells become neurons, differentiation, RNA localization in dendrites, how synaptic plasticity is controlled. These topics, my work even extends all the way to questions of brain evolution, which we'll talk about later today.
Okay.
You might think that these topics are a little bit diverse, and the thread linking them might be a little thin, but in fact, in my own mind, this is what I mean about creating an intellectual atmosphere. We operate with a lot of projects that cover a lot of ground in neuroscience.
Okay. So, and if you don't mind, can you please share your presentation?
Absolutely. Yeah, let me get that up here, and just showing the, and you should be seeing this in a moment. How about, is it up there now?
Not yet. Yeah.
Oh, there it is? Okay.
Yeah.
Okay, and is it looking okay? It's formatted?
Yeah, it looks good.
Good. Okay, so what I'd like to talk about today is right there in the title, Primate Brain Expansion, Nature's Social Experiments with Gene, Cell, and Anatomical Novelty. So I'm going to spend the first third of the presentation explaining what I mean by nature's social experiments. And then the last 2/3 will get into the weeds, the genes, cells, and the anatomy of the brain to get at the molecular underpinnings for brain expansion. And so let me go to the next slide here and show you something. Here is a phylogeny of the human lineage. We call all these creatures that were present on the earth before us and led to us, Homo sapiens.
Mm-hmm.
We refer to them as hominids. There were many of them. We don't even know. We only know a very small percentage of them. They have names like Homo erectus and Australopithecine. There's a number of many of them. But I'd like to pose the question to begin, where did they go? Because they're all extinct. The one that was present closest to us in time were the Neanderthals, and they too have gone extinct. So why were they unsuccessful? If we define the measure of success as survival, none of them survived.
Yeah.
So now this kind of, so there you can see here that over time, certainly within the hominid lineage, our relatives diversified greatly. I mean, just look at all those different individuals there that evolved within Africa, within related ecological niches. There was a lot of space to create speciation. And it's quite interesting that species are able to do this, to speciate within the different environments that they have available to them. And hominids were able to do that. The most dramatic example of this phenomenon are beetles. There are 380,000 different species of beetles, probably many more. And I'll read you this quote here from Haldane. In the company of a group of theologians, he was asked what one could conclude as to the nature of the Creator from a study of his creation. Haldane said, an inordinate fondness of beetles. It's a very famous line because beetles are so diverse. Nature created many, many of them in enormous varieties. And there were varieties of hominids too. Not as great, but still quite a few. And here's another way we can look at the hominid tree. You see at the very bottom are chimpanzees as an outgroup. And then we see this typical manner in which evolution is often displayed here, in which there are nodes and there are common ancestors from which these species that are present today, present up to the modern time, not today, but present, who derive from some ancestor back at one of those nodes. So if you look in the light purple box there, where you see Homo neanderthalensis, we see those groups that were most closely related to us. We're learning a lot about this from people like Svante Pääbo, who've been able to sequence the DNA from Neanderthals, also from another group of hominid called Denisovans. We've not been able to get DNA yet from Homo erectus, but there are others that are close to us, and what they really have in common, this group, this what we might call pre-modern Homo sapiens, is there has been a remarkable brain expansion in this group. We do know that in the hominid lineage, the brain has expanded compared to other primates, chimpanzees, but late in the hominid lineage, where this pre-modern species appear, the brain expanded yet again, and it became really quite large. And that group lies within this circle right here. And you can see on the bottom, on the x-axis, this is the number of years ago in millions in which these species were thought to be present. So this enormous expansion of the hominid brain began about 2 million years ago. 2 million years ago.
Wow.
And here's a picture of what I'm talking about. Hominid strategy of brain expansion is an expensive strategy because the brain uses a lot of energy. You're probably familiar with the fact that brain tissue will The brain uses about 20% of all the energy that we have, that we need to expend 20% of our energy budget for supporting the brain, even though the brain is only 3 pounds. In terms of weight, it's a small amount of the body, but in terms of energy, it's huge. And if you're a teenager, if you're young, that number, it's not 20% to support the brain, but closer to 50%. So the brain is using a lot of the body's energy. And what are we getting in return? What do we get in return? What is the fitness advantage of an expensive brain? So we can ask the question, what was leading this? Here was this increase. What did we get? So we can think of these as experiments in nature. And what I would like to propose here at the opening of this talk is that what nature was tinkering with in making a large brain was social behavior. Many of the experiments that were being done with these different hominids, I would speculate to say that they are related to how to create social environments, relationships among individuals that would foster survival in those groups. So the hypothesis out there, what I would like to put out there is that evolving a social structure among hominids is required for success to pay for the big brain. That is, if a hominid is able to depend on their peer groups and to have assistance in procreation, seeking food, being able to do all the things that we need to do for survival, they will more likely be successful. However, getting the social organization right is really complex. It's not so easy to understand exactly all the factors that go into getting relationships among individuals within a population to work well. And we see that today. We still contend with how we can deal with each other in a way that is going to be beneficial for the population. So, but the problem of sociality, that is the problem of species working together within a population, is very old. It's not just a hominid thing. It's been going on for a long time. So biology fundamentally requires that organisms establish a social structure. And we see that among bacteria, where we can use what are called quorums. Bacteria do what's called quorum sensing, in which they are aware of the size of the colony, and they can control the colony by sensing group behavior. Here are some snails. This was a riverbed here, in which there were many rocks that didn't have any snails, but all the snails, for some reason, liked this particular rock. Another example here with these meerkats all enjoying each other's company. And I've always wondered, look at just birds on a wire. They like to cluster. Many other wires are empty, but here they all are hanging out together. Species like to be with each other, most of them. What's that all about? What are the genes involved that control that? Well, an extreme example of social behavior is called eusocial behavior. But before we get to that, let's discuss a phenomenon that's more common, that's on the way to eusocial behavior, something that is not quite as radical, what we would call pro-social behavior. And here's the definition: voluntary social behavior intended to benefit unrelated individuals or society. That is, groups in which there's division of labor, trade, armies to defend whole groups of people, care for the needy. All of this regulated by shared moral systems that are enforced, albeit imperfectly, by third-party sanctions. So this is the way in which we protect each other. And we really do, even when people die, that we do in cemeteries. So we could ask the question, what's the motivation for this? And many psychologists and philosophers, we're not going to talk much about this today, have ascribed different types of motivation, empathy, concern for the welfare and rights of others. Maybe there's egotistical reasons to do this because we defend others, we might get some credit. There are practical concerns such as self-interest. We might help others because we expect reciprocity. And altruism, in its highest form, is an unselfish interest in helping another person. So all of these have been discussed and by others, and we won't go too deeply into that. So eusocial behavior is an extreme form of social behavior in which the cooperative brood care, including offspring from other individuals and a biologically specialized division of labor, so-called caste, exist in reproduction, and therefore there are sterile groups. That is, individuals lose their individuality. Some of them become sterile and just have no offspring, but they are there to serve the colony. And in this way, we see that in some ways, a colony, a eusocial colony, is behaving the way cells do in our body. Each one of our individual cells has given up its individuality, has given up its independence to serve the organism. Mm-hmm. Nature has taken that to an extreme example in this eusocial behavior where some of the creatures in the colony cannot survive independently, have given up their independent existence to serve the greater good in the colony. An extreme example. So, that sociality has evolved independently in nature many times. It's something that nature has come back to, and you can look at this slide perhaps later to look at all the examples of it. But let's get back to the human condition and how in the hominid lineage, the human hominids are dealing with social behavior. Let's think about that a little bit. So humans that are on the earth today arrived here in what's called bottleneck. That is, there was a catastrophic event of some sort. We don't know all the details about this, in which the proto-human population dwindled maybe as few as 15,000 individuals about 1 million years ago. So this is before Homo sapiens, but it was a time when hominid populations were really, really small. And then in the as the species that were on the tree that branched closer and closer to Homo sapiens, perhaps our more immediate cousins, there began to be a dispersion of these hominids. That is, individuals from these groups began to migrate around the world. Neanderthals, who left Africa long before humans did, went to Europe, went to Asia. And then about 100,000 years ago, when Homo sapiens emerged in Africa, our ancestors also left that continent. But in the case of Homo sapiens, we traveled really vast distances. I mean, within a few tens of thousands of years, Homo sapiens populated the entire planet.
Yeah.
It was really extraordinary. The speed at which Homo sapiens was able to adapt, as you can see here, to leave Africa, to cross all through Asia, to go across to land in the Bering Strait, just crossing at a time when there was really an ice age in this area, through unbelievably difficult terrain, and to migrate down the coast, the west coast of North America, down through Central America and South America. Other branches went to Australia. Other branches turned left when they left Africa and populated Europe. In a relatively short time, Homo sapiens really not only covered the planet, but something about the curiosity of our species would look out at some uncrossable vast terrain and say, I want to go there. What drove them? Was it curiosity? Was it famine? What made people distribute themselves so broadly across the globe, more than any other species that we know about in the hominid lineage? So now here's a dramatic picture because I do want to point out here what a cooperative species we are. We do have pro-social behavior. This is an event that happened in Utah, I believe, a few years ago, where got a lot of press attention for a day or two, in which a car ran into a motorcycle here. The individual in the motorcycle was underneath the car, and the motorcycle caught fire. It was witnessed by a number of people. This woman that you see up in the upper left got down and looked under the car and spotted the individual. She then called together the people around her who spontaneously, there's no rehearsal here. These people spontaneously got together. You can see each one of these images, and they cooperatively, with no pre-planning whatsoever, lined themselves up, lifted up the car, pulled out the individual who's right here, and he did survive. Humans have this ability to cooperate. Of course, we're not unique for that, but I think it's an amazing feature. But our ability to cooperate is very varied across our population as individuals. Here you see a network of scientists, and the way this network is linked, the edges that connect the nodes here, are based on cooperativity. So you see three different colors. These are publications published together, having grants together, and are doing many different activities together in green. These are the three colors up here. And you can see that in the center, there's a very large group of scientists that are working with each other. They share authorships, they share grants. Scientists are actually surprisingly social creatures. And we like to work together, most of us. If you look out in the periphery here, there are isolates. There are individuals that are working with maybe one other individual or no other individuals that are just not connected hardly at all. And so that human behavior, and this is one of many examples I could give you, really operates over a very wide axis of sociality. Most of us are quite social, a few of us are not. It's a distribution. And there are natural experiments that have gone on in the human lineage that affect this social axis. So here's one natural experiment. There's a condition, we're all familiar with it, autism, in which the most prominent impairment is an impairment in social behavior.
Yep.
Right here. On the other end of the spectrum is a syndrome known as Williams syndrome, in which these individuals also have intellectual impairment very often, like autism patients do. They may not be, their IQ is low. But in contrast to the autism patients, these individuals with Williams syndrome are hypersocial. You're in a crowd with a bunch of kids that have Williams syndrome. They are unafraid of adults. They make eye contact. They cluster around strangers. They love to talk to people. They are really the complete opposite of autism. Thus, nature, even among us, has genetically experimented with an axis that involves two extremes of social behavior: relative social isolation in autism and hypersocial behavior in Williams syndrome. So let's think about how we can study this. And I say this is a natural experiment because in Williams syndrome, we actually know what the genetic basis of this syndrome is. There are 25 genes that are deleted, 25 contiguous genes that are deleted in this syndrome that lead to the phenotype. And in autism, we don't know nearly as much about the genetics of autism, but there is one rare form of autism in which the very same 25 genes that are deleted in Williams syndrome are actually duplicated in this unusual form of autism spectrum disorder. So we have here an axis that goes quite symmetrically in these two directions, with a gene deletion resulting in hypersociality and a gene duplication resulting in impaired sociality. And these extremes can tell us a lot about what's connected to social behavior. When we think about social behavior, it's not just how well we get along with each other. People with autism are often have what's called social overload, sensory overload. They have a difficult time processing many stimuli simultaneously. It's very, very hard to do that. You might not think it's immediately obvious that that should be related to social behavior, but social behavior does require that we are computing many inputs simultaneously. We're listening to a person, we're watching a person, we have a lot of things we have to block out around us if we want to pay attention to the other person. When you are unable to filter sensory information, social behavior is impaired. Autism individuals also have a certain rigidity or require structure in their behavior. Here's an autistic child who's fallen asleep after lining up his toys in a perfect line, a feature that you often see in autistic children, this kind of demand for order and structure. On the other hand, autistic individuals have these islands of excellence. They often have unbelievable memories, especially high-functioning autistic individuals, what used to be called Asperger's syndrome. Some of them have artistic excellence. These exceptional abilities exist in islands, and in some ways, for these individuals, pay the price to have this ability, these unbelievable memories, by having impaired social behavior. Now, Williams syndrome has a number of features that include, I mentioned, intellectual disability, this typical facial appearance that you see here. There are other disabilities with anxiety and phobias. Sometimes they also have these remarkable musical abilities. That's also true, but most dramatically, they have engaging personalities. They take an extreme interest in other people. And they have one other feature, which is even though their intelligence is impaired, their language is surprisingly intact. And a very famous example about this, presented by Ursula Bellucci, a very famous example, is when a Williams syndrome child was asked to draw an elephant. This is what she drew. You can see that this child, who was already old enough to do much better than this, when asked to draw an elephant, really sketched something out that is hardly interpretable as an elephant. This kid's drawing ability is really bad. But when asked to describe this picture, listen to what she says. What an elephant is, it is one of the animals. And what an elephant does, it lives in the jungle. It can also live in the zoo. And what it has long gray ears, fan ears, ears that can blow in the wind. There's a long trunk that can pick up grass or pick up hay. If they're in a bad mood, it can be terrible. If the elephant gets mad, it could stomp. It could charge. Sometimes elephants can charge. They have big, long tusks. They can damage a car. It could be dangerous. When they're in a pinch, when they're in a bad mood, it can be terrible. You don't want an elephant as a pet. You want a cat or a dog or a bird. That's remarkable. I mean, this child with pretty severe impairments, cognitive impairments, is using words that are almost poetic, fan years. I mean, that's really quite remarkable to be able to write like that. Well, in Williams syndrome, we know that there are 25 genes that are deleted. You can see them on when you do a chromosomal spread, and you can actually find the site where those genes are deleted. And we know what those genes are. So we can really get some big insights here into social behavior. And that kind of work is just beginning to emerge. I'm not going to talk about too much more about that today. We've identified a gene in that group called BAS1B, which we believe is very important for some of this social behavior. Other investigators have found other genes within that group that are probably equally important, and this is an area that is rich for future research. So I think what I would like to do here is now to jump ahead and just finish up this segment. I'm sorry, I'm going to just skip a couple slides and finish up this segment with how to think about mapping genes onto behavior. And in doing this, I showed you this asymmetry here, this axis in which social behavior exists at these two extremes. And this area in the center, in this box, points out those realms that go along with the social behavior that are divergent along this axis. So theory of mind is one of them. We'll talk about that. Language, empathy, social interaction, sensory hyperawareness. I'll just mention a few of these briefly to finish up this segment. Well, one place where this axis is quite divergent is on theory of mind. This is our ability to know what other individuals are thinking. It's very important. I think most people listening to this will know what that means, but just to remind you, kid goes into the psychologist with his mother. Psychologist asks the mother to leave the room for a moment. The psychologist takes these Smarties out of the box, puts some pencils in, closes the box, brings the mother back in the room, and says to the kid, "Kid, what does your mother think is in the box?" And if the kid is under a certain age, the child will say, "My mother thinks there are pencils in the box," because the child believes that everything he or she thinks is what the other person thinks. But after around age 4 or so, 3, 4, 5, theory of mind kicks in. And now when you ask the older child what his mother thinks is in the box, they'll say, "Oh, it's a Smarties box. She must think there's Smarties, even though I know better. I know there are pencils in the box." This is the ability to know what other people think. And we acquire that, but those with autism have impairments in that ability. It's a crucial ability in social behavior. We need that to understand each other. If we don't know a little bit about what each of us is thinking, we're going to make a lot of mistakes. Of course, this can get very complicated. And we have some ability to know what the other person is thinking, but if we want to know what the other person is thinking about what yet a third person is thinking, or to go along in a chain the way poker players do, like I'm holding a hand here, and I wonder what this dog is thinking that dog is thinking about my hand. As you go up these levels, we lose that ability very, very quickly. So our theory of mind extends to the other person, not far beyond. Language is a social module. I mean, this is something that is showing up on this axis. Very often, those with autism have impairments of language. Those with Williams syndrome have these hyper abilities with language. But social behavior in hominids has been around for a while. This is what I want to talk about, this experimentation. So sometimes language, what we do with language is actually not just communicate. there's a term that's called phatic expression down here. You can see it. This means that the only function of this type of language is to perform a social task. It's the kind of thing where we don't really care about the answer. We're just talking to somebody, talking about the weather, and we say, how are you? We don't care really that much how they are. We just want to make conversation, chit chat. It's very similar to grooming in other primates. We're using language for phatic expression, not for communication. And in fact, probably most of what we do in language is more in this realm than it is actually in the realm of communication. We can effectively communicate in many different ways, but language really helps us form relationships. And that is the phatic aspect of language, which exists as part of a social module. Well, I mentioned this sensory hyperawareness, and here in autism spectrum disorders, it's pretty intense. There's also a balance here. I put a quote here actually from Marcel Proust. As you know, Marcel Proust has impressed many neuroscientists by his insights into the human mind. And here's a quote: "An hour is not merely an hour. It is a vase full of scents and sounds and projects and climates." Is that a kind of sensory hyperawareness? I think it is. So that people in literature have been able to use sensory hyperawareness actually to create enhanced reality and intensity of the environment. It's something that we want to have some balance with regard to sensory hyperawareness, not to have it be intrusive, but to have it be a part of our lives. So I will now go to this slide, and at this point, I'm going to ask you to sort of take a breather. If you want to pause this for a moment, this is a good time to do it, because I'm going to change topics now.
Okay.
And get into the some ideas about the molecular basis for brain language.
Yeah. Can I ask a couple of questions on the
Absolutely. Sure, this is a good time to do that.
Yeah. Like you mentioned, there are about 25 genes, right?
Yes.
So like, are each of genes are so important, or like it is only some of them which are important, others are not? Like it is just and people haven't identified all of them, I guess.
Most of them have been identified.
Okay, but function is not known, maybe.
But you're right. The function and some of them even we know a bit about their functions.
Okay.
Some of the ones that are, I think you're right in the fact that there is probably some contribution to the phenotype from many of them. But there are some that are particularly interesting with regard to the neurological phenotype. And those tend to be some transcription factors. The one that we're interested in, BAS1B, is a transcription factor. And we know a lot about the genes that it's controlling by doing a what's called a ChIP-seq experiment. We can see where the transcription factor is binding. And that way we can see all the genes that get modulated by its expression. And we believe that there are downstream cascades regulated by this transcription factor and probably others in the Williams syndrome locus that are important for the phenotype.
Okay.
Good. Anything else?
Yeah, that's it.
Good. Okay. Well, thank you. So I will now proceed, but as the moderator, please jump in if it starts to go too long, because I do have some more slides here that could go a little while.
Yeah, go ahead. You have another 15-20 minutes. You can
talk. We'll see how far we get, and let's see what happens. So we'll go back now. So we're very much going to change course here. We're talking now. We're going to start at the same starting point. That is this brain expansion, and let's begin by looking at this slide here, where sort of looking exactly where this expansion occurred. Well, here is an expansion of a part of the brain that here's where most of the expansion has occurred at the microscopic level. That is, it's occurred within the supragranular layer. Those are the outer layers of the cortex. I'm sure you'll recall that the cerebral cortex is made up of 6 layers. And these outer layers here are called the supragranular layers, and they've expanded quite a bit. So as a proportion of the thickness of the supragranular layer relative to the rest of the brain, primates have a much higher proportion of supragranular space than carnivores or rodents. So that may account for some of this expansion. But we still want to know if that really can account for all of it, or if it does account for it, how does it arise? It really becomes a developmental question, and that's what I really want to focus on. You could ask the question, well, if the brain has expanded, we know it has, is that expansion just same-o, more of the same stuff? Or is there a new thing that has actually evolved that takes up the space? Is there something new that's in there? And it turns out that that the expansion of the supragranular layer is an enlargement of something that existed before. But there's something else that's extraordinarily interesting. Something else of a new thing, a totally new thing that appeared within primates. Okay. And that is a developmental structure that you see right here. See these letters, OSVZ, that stands for the outer subventricular zone. And the arrow points to where it is. It's a structure that has begun to appear with delineated borders in primates. There are vestiges of this that we see in carnivores, but as a discrete structure, it's present pretty much exclusively in primates. A new thing. So to break down this picture a little bit, here's the developing mouse brain, and here's the developing macaque brain, primate brain. And to break it down, you see here there's some layers down at the bottom. This is the developing brain. So you can't think of this any longer as the 6-layered cortex. This is a whole different type of layering, because most of these cells are stem cells. These are germinal cells. These are the cells that are going to make the neurons at some point. And at the very bottom down here in the macaque, as well as up in the mouse, there are the VZ, or the ventricular layer, where new brain cells are being born, and they're migrating out. And they migrate out all the way up to the top and form, see that those letters, CP? That's the cortical plate. And this is where the 6-layer cortex is going to form, right in here. But the new thing is this guy, the OSVZ. What the OSVZ is a second source of germinal cells. A whole new location where brain cells are being born and migrating out again to the cortex and feeding the cortex, and probably feeding the supragranular layer, and may be responsible for the expansion of the supragranular layer. That's still a little speculative, but I think most people would hypothesize that's probably the most likely thing. So here's a little higher power of that sketch, where we have this OSVZ here, a major proliferative site. And as development occurs, there's a switch in which most, actually I shouldn't say, well, this is true, as development occurs, but also over the time course of evolution, there's a switch in which the major contributions of new cells to the cortex come largely from the OSVZ, and the number that derive from the VZ, the ventricular zone, begins to diminish over developmental time, and in fact, probably over evolutionary time as well. So what happened? What happened in which that we ended up with an OSVZ? How did we get an OSVZ? How did we primates get an OSVZ? So let's take a look at the time course here. We have to go back to the little bit of the geology. And you can see here are the eras in which geologists talk about the key epochs of the Earth: Cenozoic, Paleogenic, Cretaceous, Jurassic. And there is a big event, one of the biggest events in the history of the Earth, that geologists talk about a lot. It's the Cretaceous-Paleogene extinction event. This is when many of the species on this planet went extinct. It was thought to perhaps, I'm sure you've heard about this, some people think it had to do with a large comet crashing into the Earth, changing the climate. Dinosaurs here went extinct. But some species survived across this extinction event. The first primates appeared about 65 to 85 million years ago, somewhere right here, just at a time that's right before this extinction event. And they survived across that boundary. So that many of the other species on the planet, including the dinosaurs, went extinct. And something about the opening of evolutionary possibilities allowed primates to now expand. They might not have been able to do so if they had so much competition from the dinosaurs. But given this event, the impact of an extraterrestrial object hitting the Earth, now allowed for, we think, it's just a speculation, allowed for the primate lineage to expand. And this kinds of thing, these chance events that happen in evolution, genetic drift, are very powerful drivers of evolution. Well, now I want to talk to you about what we're going to study to get at the bottom, molecular basis for everything I'm talking about, or at least one molecular basis, because there are lots of ways to get into this problem. But I want to introduce this slide by pointing out that the genome has a certain number of about 25,000, 20,000 coding genes, and those genes make up a relatively small proportion of the 3 billion nucleotides in the genome. They're really about, some people say maybe 3% of the whole genome. Small percent, but we've increasingly over the last decade or more become aware of a large portion of the genome that's called the non-coding portion of the genome.
Yep.
And this is where the action is, because those 20,000 genes have been around, most of them have been around throughout the entire animal evolution. They haven't changed so much. They've changed, but not as much as the non-coding part of the genome. The non-coding part of the genome is changing dramatically. This is where the evolutionary action is, that molecular evolution is not happening as much through the invention of new genes, but operating in a way in which the control over those genes, the molecular wiring of those genes, the circuitry of the genes, as it's regulated by the non-coding portion of the genome, is responsible mostly for organismal complexity and evolutionary change. So what you see here is a picture of one type of non-coding gene. We're going to talk a bit about this. These are a type of non-coding genes called a microRNA. And microRNAs are about 21 nucleotides long. They obviously don't code any protein. What they do is they will bind to a messenger RNA and either destroy it or inhibit its translation. And that is, remain attached to it and inhibit its translation, or destroy it, which obviously also inhibits its translation. So microRNAs control whether or not mRNAs will make a protein. And this non-coding part of the genome, now we can turn to this slide, you can see how the number of microRNAs over evolutionary history has increased dramatically. So if you go to the very top, in these species up here, the sponge, these are sponges, at the very basal organisms in the animal kingdom, the sponge has about 8 microRNAs. If you go all the way down to humans, humans have probably, well, maybe a minimum of 1,000 microRNAs. There's still a lot of controversy about exactly how many, but somewhere in the range of 1,000 to 2,000 microRNAs. MicroRNAs have increased with organismal complexity as you go down this list vertically. That is, very often, once microRNAs are invented, they're not often lost. They're highly conserved. They usually stay within the genome and increase in their numbers. So microRNAs are a good candidate for thinking about maybe the evolution of new structures like an OSVZ. Maybe they were somehow contributing to that. So I'm going to skip that slide in the interest of time and go right to this. And now talk to you about the experiment that we did. So in collaboration with a group in France, the prime principal investigator there was Colette Dehaye, and with a graduate student in my lab named Marie-Luz Arcila, we, and this part of it was done by Colette, she laser dissected a region of the developing macaque brain here. This is the visual area, area 18 and area 17. This is the region in yellow that she cut out. And here are the layers that you saw a few moments ago. Here's the OSVZ here, the cortical plate where the mature neurons are. And down here at the bottom is the ventricular zone, a very ancient place where germinal cells also live. And we laser captured those, and we then sequenced. We did deep sequencing of the microRNAs. And we found in those tissues 766 microRNA precursors. Remember, microRNAs, there are only 21 nucleotides, but they come from a larger precursor structure. It's about 70 nucleotides and forms a stem loop. So there were these precursors, and stem loops have 2 arms. The stem part has 2 sides to it, and either side can potentially make a microRNA. So you double the number, and you get that there are 1,532 possible microRNAs that we sequenced. But we wanted to make sure that there were enough there to be meaningful, so we filtered them to be sure there were at least 5 counts per million in at least 3 of the libraries. Remember from the previous slide, we did microRNAs from many regions: cortical plate, the OSVZ, the VZ, and we made libraries from each of those regions. And after all of that, we found 752 microRNAs that remained. That looked like they were pretty solid, pretty strong candidates to actually be microRNAs. Well, 321 of them were expressed in primates, but they weren't reported at all in any rodents. Now, maybe some of them have been found subsequently, but look at the difference here. This would suggest that when evolution, as with the emergence of primates, a very large number of microRNAs also emerged. What are they doing? Well, that we became very interested in that. So we looked at how they clustered here on a principal component analysis, and we can cluster them by where they actually are expressed, so that these microRNAs that have high levels of expression tend to, one group that has high levels of expression tend to cluster among the cortical plate, and another group tend to cluster here among in the germinal zones, the OSVZ and the VZ. Here are the initials. And we can cluster them again in different ways here. We can cluster them in subgroups. And what you ultimately get to after looking at this slide in a little bit more detail is that microRNAs turn out to be a very good readout to tell you what region of the brain you're in the developing brain. Because here's a cluster that looks quite distinctive for the OSVZ from area 17, another one from OSVZ in area 18. So the variation, the collective variation, which is what a PCA, a principal component analysis, tells you, the collective variation of these microRNAs, when you look at them together, are actually indicative of the anatomical location. They're telling you something about the anatomy, their expression patterns. So here we showed here doing a heat map, those microRNAs that are expressed in these different regions. Here's the cortical plate, the OSVZ, and the VZ along this axis. You can see right away what was already pretty clear, that the cortical plate seems to have a differential expression clustered differently than the germinal zones here, but a very large percentage of them. See, these are microRNAs in which we don't really know what their targets are. Just to remind you, the microRNA will target an mRNA, and it will then control its expression in some way. So what are the mRNAs that these microRNAs are targeting? Some of them we know. Some of them are targeting the cell cycle or apoptosis. They're genes that are under the control of microRNAs during differentiation of the brain. But there's a bunch of them that we don't really know what the targets are at all. And many of them are, as we look down here, this is the set that's not expressed in the mouse. So many of them have unknown targets, and they're uniquely expressed in the primate, not in the mouse. So here, again, I think in the interest of time, I'll leave this slide here so it'll be available to look at. I'm going to go to the next slide. And we then wanted to go more deeply and figure out what are those microRNAs that were invented in primates. What are they targeting? What are they controlling? Now remember, genes are old, microRNAs are new. These microRNAs were invented in primates, but most of the genes have been around for a long time. So these, we expect to find, and we did find, genes that have been around for a long time in animal lineages, in which are now being controlled in novel ways. And one of the most interesting ones that we found, we used a technique here. It's called a risk trap. Words right here. It was invented by a postdoc in Dick Goodman's lab, named Lulu Cambrone, who we collaborated with. And you can fish out these targets using a risk trap method. Won't go into the details. And when we found those targets, some of the genes that turned out to be targeted by microRNAs invented in primates are listed right here. One of them is MLL, histone lysine N-methyltransferase 2A. Now, why is that interesting? Well, what this gene does, this is a gene that creates epigenetic controls. Epigenetics is going to be a very important factor here. And while we talk a lot about epigenetics, I mean, fundamentally, epigenetics still rests on genetics. So when we introduce novel epigenetic controls, and this enzyme is an epigenetic control element, we are now able to rewire, control in novel ways in which genes are expressed, because that's what these genes do. So this one, this histone lysine methyltransferase, as you know, one of the epigenetic controls is histones will control the gene expression by how chromatin is arranged. What happens is that when you modify lysine on the histone, you can control whether or not that histone will affect gene expression. And in this case, here is a enzyme that's putting the lysines on the histones and controlling gene expression. So this is one of the control elements that has been around for a long time, MLL, that now is regulated in a novel way in primates. And a cofactor that works with MLL called cancel-1 is also a target of a different microRNA invented in primates. Cancel-1 actually is known to, when it's deleted, there is severe mental retardation here in this syndrome, just to point out a little background. Well, now I'm going to come toward the end of this section. It might be a good stopping point in a moment, but let me just show you the kind of culmination of our thinking of these discoveries as we put them together. Because MLL and many of the other genes that we've found that are targeted by newly invented microRNAs are operating on the cell cycle. They're operating on whether or not a cell will decide to continue to divide, to renew, or to undergo differentiation. Well, that's what's going on in the OSVZ, in the VZ. That's exactly what's happening there. Cells are making the decision to begin the path toward differentiation. They're just like they're counting their numbers to see how many are needed so we can get a big brain of 86 billion cells. And all of these computations that have to be done, the groundwork for all of that is happening in the stem cells, the neural precursor cells of the brain, with determinants over the cell cycle. And the fact that the newly invented micros are actually targeting a cellular phenomena, the cell cycle, which is as old as the hills, is just showing how much evolutionary space remains in these very ancient cellular systems. Enough space, enough evolutionary space to create this brain expansion that occurred late in the hominid lineage. And so what this picture shows here are the cell cycle laid out linearly. You recall that cell cycle involves a phase called G1 where cells can exit and begin to differentiate, or they can continue and replicate their DNA. That's the S phase for synthesis of DNA. They can go into another phase called G2, and then they mitose, they divide, and then they cycle around to G1. So it's just spread out here linearly. And we already know, as shown on this picture, there are many microRNAs that target cell cycle genes. For instance, here's MIR1224 targeting cyclin D, 137, cyclin D. There are many examples. But now, in this slide, what I want to show you in red are the novel primate microRNAs that have been introduced in hominid evolution that also superimpose their control on top of pre-existing controls to regulate the cell cycle. And this is the kind of complexity that we're talking about that is probably closely related to what happens in the OSVZ and is responsible perhaps for its, or contributes, I should say, to its evolution. So I will just conclude here by pointing out that this portion of the cell cycle where MLL and other genes work, making these decisions, is an evolutionary hotspot. And I will then finish the entire presentation here by coming full circle here and asking the question that we started out with about this experiment that's going on right now among hominids, and seeing if these molecular changes with the microRNAs, the OSVZ, the different hominids that existed on this planet, will humans get it right? You know, we've only been around for 100,000 years. Neanderthals had a much better run than we did. They were around for 300,000 years. But there have been repeated hominid extinctions. Maybe our social organizations are just not adaptive for long-term success, especially for supporting these costly brains. Might we evolve tools that will solve these problems? The way to evolve tools by evolution is very slow. But maybe there are non-genetic ways through perhaps the various electronics and internet ways that we are now working on to actually help our evolutionary, help our survival in an evolutionary manner. So let me just tell you, here are, I'm fortunate to live in Santa Barbara and in a very beautiful place. There were many collaborators I had for this work, although one of the most important was Colette DeHay. You can see her name here. Richard Goodman, who helped with the risk trap. Additional work that I didn't talk about from collaborators at UCSF. And finally, a picture of my lab. The work that was done with the microRNAs was done by this individual right here, Mary Arcila. She had a bit of help from other people in the lab, including Robin Zhu and Neha, and there are others who have contributed quite a bit. Well, thank you very much.
Thank you, Dr. Kosik. It's a pretty nice presentation. It's, I mean, like, I think you are opening up Pandora's box, looks like. There are a lot of things we could do. Can you please unshare the presentation?
Yes,
so
I'll
that,
go over to that. And,
yeah.
Good.
So this is an excellent, like, I mean, like, it's really thought-provoking, and people can, I mean, like, jump in and do a lot of stuff, like with the current invention of mRNA, microRNAs and all. I have one question, like you showed in one of the slides that a bunch of microRNAs which are present only in the primates and all. So has anybody tried to put them in, like, even though we are not sure what they do, put them in mice or somewhere, like, and see, like, hey, what happens? Do you see any behavioral changes or anything like that?
We haven't done that.
Okay.
You're trying to humanize a mouse?
Yeah, no, I mean, like, just to say, like, what happens, right? Like,
Yeah.
quick and dirty experiment trying to see.
Yeah. We did, I didn't have time to talk about, we have done that. There's another category of non-coding RNA called a link RNA that we've also discovered that is something invented in the hominid lineage. And we have done those experiments for that one. And we can show that it will alter development in a way that is interesting with regard to exactly the question you're asking.
Yeah. And OVSG, like, what is the, like, why it is so that it happens in there? Like, what is the, like, what happened? Why it is only fixed in that area rather than somewhere else? Is there any particular relevance to that area?
When you say that area, because the OSVZ is found throughout the developing brain.
Yeah, yeah.
And supplies the cortex. But when you say that area, do you mean?
Yeah, where you have these like stem cells growing, right? Like, why do you have that area rather than somewhere else? You know what I mean?
Well, so that's a good question. There's a long evolutionary history in which nature has, long before primates, in which nature has used a region of cells right next to the ventricle as a niche for stem cells to supply the brain.
Yeah.
And evolution likes to build on what already existed. Yeah. So by expanding that layer that had pre-existed and making it larger, separating it into a distinct layer, giving it new properties, inventing microRNAs that operate within there, changing the kinetics of division, affecting the cell cycle, by changing things that were already existing, you have actually, I think, a key way, insight into a key way in which evolution works, which basically is that evolution basically deals with constancy and change. And it has a very interesting dance between those two phenomena, keeping things the same and yet having enough room to change.
Okay, that is interesting. Yeah, like, it makes sense that rather than doing it all together differently, starts from where it is there, then modify it and make minor changes with the time so that it helps. Okay. And like these microRNAs, right? Like they are just, I mean, like, no, still there are, even though every day one new microRNA gets invented, looks like. But I think like in addition to this, like controlling RNAs and all those things, do you see any other role? Like, I mean, like everybody is hitting the same target. Has anybody looked into, okay, it might be doing something totally different than what everybody else is doing?
Well, it does seem that the main job of microRNAs is a post-transcriptional regulator, and they do that by affecting mRNA translation. If they have some other function, I think that's still there's been debate about that, whether they might operate in the nucleus. It has not been very well substantiated. Most scientists believe that microRNAs are working almost exclusively on the mRNAs, but that gives them a lot of room to play. I mean, the way to do that is very complex.
Yep. Okay. Yeah. Thank you very much. I really appreciate you coming on to Science Hangout and sharing your work with us. So again, from Science Mission, really thank you from the bottom of our hearts to make this presentation possible.
Thank you very much for having me on. Right.
Thank you.